How does CRISPR gene editing use homology-directed repair? Imagine a tiny cut in the DNA of a living cell, like a microscopic scratch on a piece of fabric. That's what happens when CRISPR gene editing makes a precise cut in the DNA. The tool used is usually a protein called Cas9, which acts like molecular scissors. But the real magic happens after the cut. The cell has to repair this break, and it can do it in a couple of ways. One way is called non-homologous end joining, which is like taping the torn fabric back together. It's quick but often leaves mistakes, leading to random mutations. The other way is homology-directed repair, which is more like following a detailed blueprint to fix the fabric perfectly. Homology-directed repair is a natural process that cells use to fix DNA damage. But in most normal cells, it happens less often than non-homologous end joining. That's why scientists have to get creative. They introduce a piece of DNA that acts as a template at the same time as the CRISPR cut. The cell then uses this template to repair the break, copying the new sequence into its DNA. This allows scientists to make very specific changes, like correcting a genetic mutation or inserting a new gene. However, homology-directed repair mostly works when cells are dividing. When cells are in the process of copying their DNA, the repair machinery is more active. To improve the chances of successful editing, researchers try different tricks. They time the CRISPR cut to happen during cell division. They also attach the Cas9 protein to other proteins that only work when the cell is in certain stages of its cycle. Some scientists even use light to trigger the cut at just the right moment. Sometimes, despite all these efforts, the cell still prefers the quick fix of non-homologous end joining. Other times, the repair works perfectly, but only in a small number of cells. And occasionally, the cell does something totally unexpected, like inserting the template in the wrong place or creating strange rearrangements. These surprises are part of what makes working with CRISPR so fascinating. In real-world applications, homology-directed repair is used to fix genetic diseases, add fluorescent markers to see how proteins behave, or even change entire genes. Some experiments have created animals that glow in the dark or bacteria that produce useful materials like spider silk. Because HDR is tricky, scientists often combine clever biology techniques with a bit of luck to get the best results. So, in simple terms, CRISPR uses homology-directed repair by making a targeted cut and then encouraging the cell to use a supplied template to fix the break precisely. It's a mix of high-tech tools and biological chance, making it one of the most exciting and quirky areas of modern science.